This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV. This Week in Virology, episode 377. Today is February 19th, 2016. Hi, everybody. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Good afternoon.
So it's 60 degrees Fahrenheit.
Oh, no. How is it
16
possible?
Celsius. Yeah. Or another app says it's only 53 Fahrenheit, but it's way up there.
It's 2 degrees Celsius here. And totally cloudy and supposed to get some kind of precipitation tomorrow. It got, it's weird. It got warm at the beginning of the week. Now it's cold again. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's a very exciting time weather-wise here in Western Massachusetts. We're clearly just about to enter mud season.
Is it raining there?
It's clouding over. We're supposed to get snow, then rain, snow, and then rain, snow, and all this, switch back and forth between the two precipitation types and highs in the 50s, and that's when everything melts and it gets really fun around here.
And you put away your wovel because you can't do anything with mud.
Because you can't shovel any. No, it's useless to try and shovel the mud.
Also joining us from North Central Florida, Rich Condit.
Hi, everybody. Here we are. You want to hear the weather?
Waiting with bated breath.
We got 71 degrees, mostly sunny. That's 21C for you Celsius dudes out there. It's a glorious day. This is a great time of year. Great time of year. It's beautiful.
Today we have a half Zika episode. But you're not going to know which half it is until you listen a little more.
The intention was not necessarily to have a half a Zika episode, right?
No,
but stuff just keeps pouring in.
Yeah, and it's kind of interesting. So,
Yeah.
But we
And I think we're going to kind of do probably what we did with Ebola, where we eventually get to the point where we're just doing a Zika sit rep.
Yes, exactly. Yeah. But we need some more data, so we'll do that.
Yes.
But we do have a paper on a different virus today, and that's very exciting. So listen for that. But in the meantime, I need to tell you that this episode of TWiV is sponsored by the 32nd Clinical Virology Symposium by the American Society for Microbiology. Now, next year will be the 33rd, and there will not be any confusion about the length of the meeting, right?
Right.
You can now send in your abstract. You have until March 17th, which is just about a month from now, to attend this meeting, which is happening from May 19th through the 22nd in Daytona Beach, Florida. And at this meeting, you can talk about your work on rapid viral diagnosis, clinical course of viral infections, and preventing and treating virus infections. There will be over 1,000 people there, including basic scientists and primary care physicians. If you want more information, go to asm.org/CVS2016. We thank ASM for their support of TWiV. Now, as I mentioned a couple of weeks ago, we are looking for a laboratory technician here in New York City, and you can learn to work with a variety of viruses. And now I do have a web page to send you to, and I'm going to put a link to the show notes so you can see a description of the position. So if you're here in the New York City area, you could apply for it and possibly come and see the wall of polio.
Yeah.
Got any applicants yet?
We do. We have about 20.
Whoa!
And, the ones that have driven ambulances, that's not what we're looking for. We do have a few people with some lab experience. So
did you get any from your TWiV announcement last week?
I wouldn't know, because I just said we're looking for someone. So I don't know. I didn't have a website, so now we do. So we'll see if some come in through that. But 2 years or so where you can learn virology and contribute. And what viruses? Well, you've heard them all here on TWiV.
That's right.
All right, we have some follow-up from our friend Johnny in Cambridge. writes, please see particularly the section Approach to the Infant with Suspected Intrauterine Infection. So she sends a message, which I think was forwarded to her, and it has a link to an overview of torch infections, which is in a publication called Up to Date. This is for docs, I guess.
Yeah, cool publication.
Yeah, there's a lot of information here. It's very nice. And thumbnail sketch about torch infections. Reaching back from my newborn medicine pediatric point of view, torch titers were drawn when an infant was born with intrauterine growth retardation, or sick and appearing septic and blood cultures are negative, or perhaps infant sick and small for gestational age, or if there were some stigmata of intrauterine infection, or in cases where the placenta appeared abnormal or suggested intrauterine infection. From the obstetrical point of view, I imagine if intrauterine growth diminished or decelerated, perhaps torch titers would be ordered as part of a workup. OB and neonatology TWiV listeners could comment more authoritatively. I don't know, do we have any?
Probably.
We'll find out.
They're silent. And she also sent in a second email, a separate link, which is rather slow. The JAMA. Oh, yes. From JAMA Pediatrics, what pediatricians and other clinicians should know about Zika virus. So this is the 1 or 2 page summary. If you don't want to listen to 8 hours of TWiV programming.
Why wouldn't you?
Yeah. Well, actually, we have 3 Zika episodes so far, right?
Mm-hmm.
It's about 6 hours of programming, but it's probably actually 5 with all the weather and stuff, right?
Well, and the first one was really only a half episode, too. Yeah. We did something else.
But it'll add up, I'm sure.
Yes.
Kathy, can you take the next one?
The next one is from Chris, who writes, the comparison with congenital rubella syndrome was awesome. Do not forget the devastation caused by other diseases spread by the same mosquito, dengue and yellow fever. The morbidity and mortality numbers for those make the stats for Zika seem very tiny. The Aedes aegypti mosquito is not native to the Americas, so any method to eradicate them, including genetic modification, is fine by me. I have had dengue fever. I don't want it again. I vote for any way to get rid of the little beasts. And this is also still from Chris?
Mm-hmm.
Okay. Dear TWiVers, please refer to the previous weather report by Barbara, since we are in the same city. It is February in Seattle, therefore it is either raining or it is foggy. My outerwear choices are either a sweater for the fog or my poncho with sleeves for the rain.
So this is actually posted on microbe.tv/TWiV for last week's episode, and there was a previous email from Barbara, who happens to be also from Seattle, suggesting a paper, which we'll do at some point in the future, so I haven't read it. So there was an interesting discussion on Twitter yesterday, and it continues today. You know, people saying, should we have Zika parties? And, I say no, because you just don't want to ever infect yourself with a wild-type virus. And people are saying, well, it's supposed to be mild. And I said, yeah, well, you don't know what the long-term sequelae are.
We used to think hepatitis B was very mild.
Yeah. And someone brought up chicken pox parties. And I said, yeah, and then you get shingles 40 years later. That's so not so nice. And then someone said, well, yellow fever is not much worse than the vaccine. I said, are you kidding me? Do you think Tyler spent years passaging yellow fever virus because it wasn't much different? No. So the wild type virus has like a 15% fatality rate.
Yeah,
right?
when they were doing the experiments at Walter Reed to demonstrate that was transmitted by mosquitoes,
they effectively
some of the people in the experiment died.
Yes.
Yeah, that's right. That's absolutely right. Yes.
Yeah,
Matter of fact, one of the experimenters died, I believe.
Yes, and that famous painting, one of those guys, they have a painting of people putting mosquitoes on a guy's arm. One of them died, yeah. One of those people was named Lazier, I think. Interesting. Anyway, that's why I brought up that discussion. This idea of having virus parties is silly. And then someone chimed in, who would bring the mosquitoes? Do you guys remember when Bill Gates released mosquitoes at a TED Talk?
Yeah.
Yes.
Alan, can you take the next one?
Sure. So Russ writes, FYI, see report of pyroperoxyphene larvicide, not Zika virus, as cause of microcephaly in Brazil. Doesn't make sense in view of the discussion on the past 2 TWiV episodes, but I pass it along, and provides a link. And this we talked about a little bit last time. And I commented, even a broken clock may be right twice a day. There are these sort of way out there folks who've been pushing this theory forward, and just because they're way out there doesn't mean they're immediately wrong. I did, however, look further into this since then, and I see, Vincent, you posted a link to a pretty good article about it as well on CBS News. So on further investigation, this is a crock. The particular pesticide, I wasn't familiar with this pyriproxyfen when we were talking last week, but this particular compound is used as a larvicide. It kills mosquito larvae. It has been tested and approved by the WHO for use in drinking water. So this is a big problem in the tropics. You have a big container of drinking water, and the mosquitoes, of course, love that because it's a sheltered pond with no fish in it, and so you get all the larvae in there. This is a compound that's put in very small quantities in those large containers and kills off the mosquito larvae. So very effective. It is non-toxic to humans. It works on a biochemical pathway that we simply don't have. And the amounts that are used in water are ridiculously below anything that could be toxic. And as this CBS article also points out, a couple of other places have pointed out, the rise in microcephaly is not limited to areas that have used this larvicide. So it doesn't even correlate.
There's a good quote at the end of the article from Tony Fauci. Says, while no theory should be rejected at this point, the discovery of virus in the brains of miscarried fetuses strongly suggests involvement of the virus.
Yes.
I think that's correct. Now,
and also pointing the finger prematurely at a mosquito larvicide could encourage people to stop using this larvicide, which would increase the mosquito population and give exactly the result that you don't want.
That's even happening in one place in Brazil. They've discontinued using larvae.
Yeah.
That's a complete disaster. And bear in mind, this is not the only virus these mosquitoes carry.
That's exactly right. Dengue and chick.
Right.
Now, I think back in December when first time we talked about Zika, remember we said, it could be something in the environment as well. We were circumspect, right? We didn't say it was the virus initially, but as the evidence slowly accumulates, you're getting evidence in favor of the virus, but we haven't concluded yet that that's it. It's just getting stronger and stronger, and there's no evidence for anything else yet. So.
And I'm not saying that it's impossible that anything in the environment is causing it, just that the case for this particular larvicide is not convincing at all. There could be something in the water, there could be something in the air, there could be some combination of factors that's going to turn out to be the case, but this is almost certainly not the answer.
Right.
Yeah, it can be Zika and something else. There could be cofactors. There's lots of.
Sure.
It pays to keep an open mind, but I agree that the evidence that Zika is a factor and probably a major factor that is gaining a lot of traction.
It is piling up. Yes.
Yeah.
Rich, would you take the next one?
Oh, goody. Martin.
With whom we have a long history.
Yes.
With whom we have a long history. He gives a link to, actually, it's a link to an article that includes this information about Brazil suspending the use of the larvicide in a particular county, I think.
Right.
He says, Dear TWiV, why don't you address the question I am trying to ask? Why are you happily part of a system that jumps the gun without proof? Fear and panic are the result, even to the point of Obama requesting $1.8 billion to find Zika. You, the Academy, are the scientific authority. You have a responsibility here. Martin, the broken clock who is right twice a day.
So let me just tell you, Rich, I've had a lot of emails about Zika from Martin. So I figured just because someone is so persistent, we could address one of them.
Yeah. I think I'm copied on a lot of those.
Yeah,
and it's the same rap. Martin, we are doing our job. we're executing our responsibility. I think we've approached this with an open mind, and I think we're looking at this objectively, and the evidence is mounting up that it's Zika virus, and the case for the larvicide is really not good. So,
but we'll see. We keep open minds. We'll keep talking about it, right?
Mm-hmm.
Yeah.
Now, a couple of things here. First, so I'm not sure Martin is actually sincere in his questioning. I think he just wants to rile us up, and that's fine.
Sure.
But he wrote us a long time ago, many times, about not believing that HIV causes AIDS because you never see virus particles in the patients. So now, last week, we reported a paper where they saw virus particles in a fetus. So you can't have a double standard. You have to be consistent. Well, anyway.
And I mean, the $1.8 billion funding, that wasn't something that I recall us specifically advocating. And against the backdrop of other government expenditures, I mean, I think we've previously picked on the military, but they're a good target for this. They are building a jet that the armed forces don't even want, and they've spent $400 billion on that. So
I'm just going to ask everyone, what do you think about $1.8 billion for Zika? I have my ideas, but I'm curious about yours.
I think that's an awful lot of money, is what I think.
Okay. Kathy, what do you think?
It sounded like a huge amount of money when I first heard it. And then I've only seen one thing that divided it out, and I didn't commit those numbers to memory, but some of the money is going to Brazil, some of it's going to mosquito control. So maybe when you divide it out that way, it's a little bit different.
It's not just Zika, basically, right?
Right. And probably there's rumors about having a Zika czar, so maybe it's going to pay that person's salary.
Oh,
we don't need another czar.
Now we have a title, Zika czar.
Zika czar.
Who was the Ebola czar? Was there one? No, I don't.
Yeah,
Who was
there
that?
was.
Who was that?
They named one.
Kathy, I think you should be the Zika czar.
Well, I think I would be the czarina, wouldn't I?
You could.
Yeah.
But,
okay,
I think Kathy has a point. I think when you divide this money up, if it's distributed, a lot of it going to public health measures and stuff as well, it could have. It's likely to have anyway, even in the research arena, an impact that goes well beyond just Zika. I mean, it's clear. We learned a lot from Ebola. And we keep learning. And these sorts of, especially in the global age here, these sorts of outbreaks are, going to keep on coming. And the more we can do to put in an infrastructure that can deal with them effectively, the better off we are. And some of this money may help with that kind of thing.
And stuff like this, as much as I hate what the budget process for science has become, it is often knee-jerk responses that get the money shaken loose from a Congress that's not otherwise willing to allocate it.
Exactly. My thoughts exactly.
So
it trickles down to other parts of virology. And
right.
science,
So yes, you've got this big newsworthy thing, and everybody wants to do something, and this is... again, I hate to put it this way, but it is an opportunity to get some money funding some projects that we should have been funding anyway.
Yeah, I agree. I totally agree. And it's not all for working on the virus and making vaccines and antivirals, as we've said. It's about other related things. There's a microcephaly issue in Brazil, and we can help.
Right. Mm-hmm. Yeah. And especially if, as Kathy says, if it's going to things like mosquito control, that's great. Yeah. That is absolutely something to put money into.
One of the important things is how the legislation, assuming there's a specific legislation associated with this, how that legislation is formulated, whether it really allows for a broad distribution of the funds, and it ought to have some of these legislations become permanent.
Right.
And wind up committing money for things that are no longer a problem. All right. And we have to be careful of that.
That's probably not a danger here. So the president has asked Congress to allocate $1.8 billion in emergency funding for this.
Right, a specific number.
Number, and so that's the president's proposal. Now, remember, the Congress that we have right now exists to not do the president's proposals on anything.
Right.
So it's whatever happens, it's going to probably be quite different from this. And I guess we'll see what comes out of it.
So I found something about how the 1.8 billion would be spent. 1.5 would go to Department of Health. 828 million for response
CDC.
Teams.
Mm-hmm.
Yeah. 250 million for the Medicaid program in Puerto Rico. 200 million for development and commercialization of a vaccine. 210 million for a fund specific to any other needs related to Zika. 335 million to USAID's efforts to support countries where the virus emerges. Final 41 million would go to the State Department to help its employees and U.S. citizens in affected countries. And I also found out that the Ebola czar left last February 15th, 2015, and it was Chuck Schumer who called for a Zika czar.
Okay.
5 days ago.
The Ebola czar was Ron McClain, who was done.
Right.
So I don't want to be cynical, but I think a lot of this is political because at the end of this Times article, Fauci says, we got caught shorthanded with Ebola before we responded, and says now we're responding quickly. So I think that's part of it. Here's something. We're not sure how important it is. So let's just jump on it and show everybody that we're on top of it. And that's
it's also, that's typical of government and even military approaches to things. Let's win the last war.
Yeah.
Which in the case of emerging viruses may not be a bad strategy. Let's figure out what went wrong last time and do it better this time.
Yep. All right. I thought that even though Martin, doesn't have good intentions, we could actually get some goodness out of that. So,
got some mileage out of that.
Thank you.
That's good.
But he's listening because he got the broken clock who was right twice a day.
Yes.
All right. A couple of Zika-related publications this past week. The first is a morbidity and mortality weekly report from the CDC. And the second author is our friend, Tyler Sharp. Mm-hmm. What was the name of that episode? How Tequila
Mosquito.
That's right. How Tequila Mosquito. And,
right, which is a pun on a method that has to do with pouring tequila on the walls of your house, as I recall.
Yes.
So Tyler got a permanent job with CDC, and he's in Puerto Rico. Is that correct?
That's correct. At the time we had him on the show, we had him on, among other things, because he blogged about a dengue outbreak in the Marshall Islands. Okay. Right. And in fact, we had him... he visited the University of Florida sometime after that, gave one of our career talks. Right. And at the time, he was an EIS officer, the Epidemiologic Intelligence Service, and he was based in Puerto Rico but taken off all over the place. And since that time, he has become a permanent staff at CDC, stationed in Puerto Rico with responsibility for dengue and other nasty diseases, and he, to some extent, credits the TWiV bump that he got from being on for getting that job.
Yeah,
of course.
So it's great.
Boss.
Of course.
Hey, you were on TWiV. Yeah, you can work for us. Well, this is a report on the first local transmission of Zika virus in Puerto Rico. And so the first locally acquired case, December 31st, 2015. An elderly gentleman of 80 years of age who had a lot of other health issues. But as far as I can tell, he doesn't seem to have traveled. So it's a locally acquired case. And then they went on and did some other screening, and they ended up with a total of 30 laboratory-confirmed Zika virus infections.
Scattered all over the island.
Yeah, so it's there, and they're going to have more.
And it's this first case that they isolated, I'm sure, is not the first case.
Yeah, probably not. It's first we know of.
First we know of, but if it's spread that widely across the island already, it's been there for a little while.
Yep. So they say at the end, we're going to follow these people and look for virus in saliva, urine, and semen to see if we can detect it. And that's good, because now you got you have the individuals initially infected, and you can follow them to see what happens.
And one of the cases they specifically mentioned was a woman diagnosed with Zika virus disease during the first trimester of pregnancy.
Yes.
Mm-hmm.
Which was in January of 2016. So
right, we'll see what happens.
That's worth watching. Yes.
Now, one of the important questions is, do you routinely find Zika in the amniotic fluid in the fetus,
yes,
or not? Is it only in kids with disease or what? And we have to find that out. It's a very important question. So,
although getting amniocentesis when there's not a specific indication,
no, it's a problem.
No.
It's a problem. But if you give birth, you could collect some amniotic fluid and just look in a healthy baby, especially in areas where Zika is endemic, right?
Right.
So now here, I may have mentioned last time I was contacted by someone locally who has a lot of people going back and forth to these infected areas in the hospital, and they want to do some diagnostics and then follow them through and see what happens in the end. So this is the sort of things that need to be done. So good job, Tyler. Very nice. The second report actually came out last Friday, but didn't make it. It's in Emerging Infectious Diseases. It's just a short note, which was sent to me by the editor there, who then said, "You didn't talk about it." I said, "Well, I'm sorry, it wasn't published." It's just a little note from the UK, and this is an individual from French Polynesia who went to the UK and was diagnosed with Zika virus infection, and so the PCR was positive on serum, but then they looked in other fluids and they found PCR positivity in semen, although they did not get infectious virus from semen. So this is someone who had recovered. Convalescent phase serum, urine, and semen samples. Only semen was positive. So that's when he was recovering, 27 and 62 days after the onset of illness.
So that interests me, okay? That because our sort of understanding currently, this may change, is that Zika, you get an acute infection and basically you're cleared within a week or two.
Yeah.
And we have, according to this, persistence of virus in semen for at least a couple of months. And, Ebola was the same kind of thing.
Right.
Where the persistence in semen went out a considerable time after one would expect that a person would have cleared the virus from all other tissues. So,
which makes me wonder if we're going to start seeing this in other viruses too, if people bother to look.
Yeah, I think it's probably right. We may not look often. Now, remember, this is a fragment, a PCR fragment. No one yet from Ebola or Zika has virus from semen. So, I mean, at some point, someone either has to get virus or the whole genome just to show that it's there, and it's not just fragments that have migrated there.
Though we do have a couple of cases that appear as if there was sexual transmission.
Yeah.
Right.
Yep. Appears, but not proven, so we'll see. Right. Yeah, this is still an ongoing story.
And if you do get whole genome, you have to do it in a way that confirms that was an intact genome.
Right.
Yeah, I'm just concerned that there are lots of pieces around that.
Sure, exactly.
Whatever, right. All right, the other is a letter in Emerging Infectious Diseases of report of Zika in Indonesia. I think this is the first report there. And so that is a very populous country with lots of mosquitoes.
And lots of travel.
So there probably will be more there. So
that's a country that's scattered across a bunch of islands, so people are constantly traveling between them and between their neighboring countries and major crossroads.
And then there's
it's amazing. This simmered for so long.
It did.
Yes.
So did HIV,
then,
though.
boom.
So did HIV.
Yeah.
Right, from 1920s to the 80s, basically. And, I mean, then you go back and you see little evidence of it here and there. But I find that really scary, this insidious simmering, and then bam. Really incredible.
We ought to get, an epidemiologist on sometime to get a perspective on the dynamics of this thing. It's like once it gets into really gets a foothold into a big population, in
a big seronegative population.
Yeah, because most of the simmering, well, there was a lot in Africa for a long time, which is not underpopulated, certainly. Maybe it's not a lot of transportation in and out of the areas where it was simmering.
I think that was it.
And then in smaller locales like, the Pacific Islands. But boy, when it hit Brazil, just boom.
Yep.
Yeah.
There's another little letter in the Emerging Infectious Diseases from this week. Phylogeny of Zika virus in the Western Hemisphere. So they compare sequences by phylogenetic trees of viruses isolated from different places. So for those of you who are interested in that, and I found a notable point of interest from this, the finding that the movement of Zika and chikungunya are strikingly similar, which I think was mentioned last week as well. But here we see it in the sequences, which is pretty neat.
So one of the things that interests me about this, so they identified basically 3 clades: West Africa, East Africa, and Asia.
Right.
And the Asian is the strain that's now circulating in South America and Asia. There's like a 10% difference in nucleotide sequence between, for example, the West African, which is really kind of the original, though the East African looks like it's a little older. At any rate, there's a 10% difference between those guys and the Asian strain. That's not a small difference.
No, it's not. Yeah.
That's a fair number of amino acids. And so I really wonder what, if any, differences there are in the biological and perhaps even serological properties of those viruses. We will find out. I suppose it's going to require, in particular for the biological properties, a good animal model.
Mm-hmm. So basically the Zikas from Guatemala and Puerto Rico, they're all Asian genotype, closely related to those in Brazil and French Polynesia. And the topology of this tree confirms the findings that the Asian genotype viruses have been gradually evolving throughout Asia since at least 1966. And the Malaysian 66 is like an ancestral genotype.
Right.
And so at some point between 47 and 66, it went from Africa to Asia.
And by the way, all these snippet papers, since they're government documents, CDC, they're all open.
All right, now...
both these journals, MMWR and Emerging Infectious Diseases, are actually great reads.
Yes.
They're really nice journals.
Yes.
So this morning, my friend, he's now my friend, Jorge Pontual, he said he listened to us last time and he laughed a lot. He's a TV news guy from Brazil who visited me a couple of weeks ago. He sent me this article in the Times. Zika may increase risk of mental illness, researchers say. And they feature Ian Lipkin and others, saying there may be other mental illness. And Pontual just basically said, why does he have to go on the who's the doctor here?
Oh,
the Oz. Why does he have to go on the Oz show and the Times and say these things to scare people? And I don't know the answer to that, but it's known that microcephaly is a risk factor for cognitive and motor delay. So this is nothing new, right?
Well, and the research tying viral infection in utero to mental illness, I mean, there's a long history there. I'm not going to say anything is settled, but there's a long history of suggestive and presumptive connections between mother's infection in pregnancy and things going wrong in the fetus that are not necessarily as extreme as microcephaly.
And we have said before, and others, we repeat what others said, that microcephaly is not likely to be the only problem in these children. So this article is just not really new, and in my view, not necessary to scare people anymore.
Well, it's hot news.
Yeah, it's hot news. That's right. Unfortunately.
So everybody wants to find a new angle on it, and what else can you say? And hey, I learned something new. Ian apparently works at a stand-up desk. So
I was just going to say that. He has something in common with Alan.
Yes, there's a picture of him here standing at his desk, which looks strikingly like me standing at my desk.
Do we need to have that picture in the New York Times, though?
Oh, why not?
It's not really news.
What a giant monster.
But let me say something. Alan, you just said We need to have something to say. We have just talked for 40 minutes on interesting stuff that has nothing to do with what's in that article. They're saying that they just don't know what's interesting. You could take basic virology and spin it so it's really cool.
Yes.
And I think that's what we try and do. Hi, Dickson.
Hello, everybody.
Hey, Dickson.
Hi, Dickson.
Hey, gang.
Now we have one more Zika paper. It's just someone just tweeted this morning. Nathan on Twitter. Thank you, Nathan. And I burned my son's omelet because I read this. And I had to make him another one, so I brought his in for lunch and I ate it. I knew it was burning, but I couldn't stop reading this article. I really do like viruses. Detection and sequencing of Zika virus from amniotic fluid of fetuses with microcephaly in Brazil, a case study. This is published in the Lancet. And this is a continuation of two earlier brief reports, which we mentioned, I think, last time, of the finding of PCR positivity in the amniotic fluid of developing fetuses who showed signs of microcephaly by sonography. So this is out of Brazil. These are physicians working all over Brazil. So what they have done here is now done a little bit more detail, because previously it was just PCR fragments. They have serum, urine, and amniotic fluid samples from both patients at week 28. Both women reported Zika-like symptoms early in pregnancy, and previously the RT-PCR showed positivity in the amniotic fluid. The urine and serum are negative. They get IgM in the amniotic fluid, IgM against Zika virus. And again, I think Carolyn was commenting on that a couple of weeks ago, right?
Yes.
That meant that the fetus was infected.
Yeah.
The torch screen was negative for all the torch pathogens. And so what they did is they took some amniotic fluid and they centrifuged it to purify virus particles and, of course, other things as well. But virus particles will go down there, and then they sequence the viral nucleic acids. And from one patient, they get the complete viral genome, which is very nice. And from the other patient, they got two larger pieces, but not the whole genome. So they can fit this into the phylogeny. But most importantly, the entire genome can be pieced together there in the amniotic fluid, right? So that means the virus has crossed the placenta, and it's in the amnion. We don't know about the fetus because these are two babies that are still alive, right? And so we don't know what's going on in them. But they say our group is the first to isolate the whole genome directly from amniotic fluid before delivery. So they say it supports the idea that the virus can go through the placenta. And listen to this. A connection between Zika virus infections and poor CNS outcomes remains presumptive and is based on a temporal association. Other possible causes or contributing factors should continue to be pursued as new cases arise. We recommend that Zika infection should be regarded as a positive causative agent in cases of microcephaly, especially during outbreaks in endemic regions. So this is really nice stuff, and it's properly conservative, I think, right?
Yes.
It's a nice paper.
I think that it was in this paper, but I can't seem to find it again, where they make the point that we discussed on a previous episode about how the Zika symptoms showed up in the mothers very many weeks before the detection of virus, in this case, in the amniotic fluid, and discussed the possibility of persistence either in the mother or in the fetus for a significant period of time, and talked about the potential role of pregnancy being mildly immunosuppressive in the mother in that phenomenon.
Yeah, so these, the rashes were at 18
so
weeks.
18 weeks, 18 and 20, two different patients.
So I presume, at the time they didn't look for Zika, and now they may have looked and it's negative, although they don't say it here, I think, right? Let's see.
Right, I don't think so.
At the time, at week 28, which is the current paper, the RT-PCR for Zika it was in the amniotic fluid, but negative in urine and serum from both patients. Okay. So at this point now, where we're seeing it in, it's negative. So yes, it seems to have come and gone.
That's an important piece of data.
So of course we don't know that the mothers were infected, right? But let's see with the antibody tests. Serology tests of serum, urine, and amnion using anti-dengue, anti-chick were negative. ELISA for Zika was positive in amnion, negative in serum and urine for both patients. So they don't even find antibodies in these two patients' sera. That's weird, right?
That is weird.
I mean, presumably they were infected because that's how the babies were infected, but that's very weird. So something's going on there. Anyway, it's another complete genome, which I think is really good and adds to the evidence. So that's cool. And that is our, what do we call that? A rapid-fire Zika review of actually 45 minutes. Dickson, you have any comments on Zika?
I heard something on NPR this morning from some Brazilian public health official who dismissed the use of this new insecticide as a possible cause and said incorrectly that until proven innocent, Zika is guilty. Well, he actually said that.
That's not right.
That's right. It isn't right.
I mean, there's
heard that.
Well, we heard what they just said in this paper. I think that's the way.
Yeah, that's actually right. But this went all over the country.
All right, so we have a paper on influenza virus, which is very, very cool. I really like this. It is published in Nature as a letter, and the authors are from the United Kingdom and France. And the first author is Jason Long, and the others are Giotis, Moncourge, Fries, Mystery, James, Morrison, Iqbal, Vignal, Skinner, and Barkley. Michael Skinner is the second to the last author, who I know from polio days, and Wendy Barkley was our guest on TWiV 177 in Dublin. Why you laugh, Dickson?
Oh, you just got a steel trap memory for these things.
I wrote it down here.
He's got notes.
I got notes. I did know that she was on TWiV, although I didn't remember that Helen Lazier was on TWiV, and that's bad. I didn't know
Michael Skinner had a polio history. He's got a pox history, too. I know him from pox.
Is that right?
Yep.
So, I still to this day feel very badly that I had forgotten that Helen was on. I should know every guest that I've had on TWiV, right?
I think that's kind of asking a lot.
No,
yeah, I think so.
You're up to
You've had a lot of
377.
I think Charlie Rose remembers every guest he's had. And if he can do it, I can do it.
He's got an assistant that just cues him in on that stuff.
So the paper's called Species Difference in ANP32A Underlies Influenza A Virus Polymerase Host Restriction. And so this is all about influenza viruses. And as everyone probably knows, one of the important natural hosts of influenza viruses are aquatic birds. And when new viruses come from birds to humans, initially they have to undergo a number of changes in receptor utilization, which we've talked about here, the stability of the virus particle, like we've learned from the Fouchier-Kawaoka experiments, and the RNA polymerase of the virus needs to have a change because the avian polymerase doesn't work well in human cells. Now, the flu virus polymerase, it's an RNA-dependent RNA polymerase. It's made up of 3 subunits, and one of them is called PB2. And every human influenza strain that we consider to have an avian origin, the PB2 has to have a very specific mutation that causes an amino acid change. And the most famous one is E627K, glutamic acid at 627 to lysine. All right. And if it doesn't have that change, it doesn't replicate well in human cells. All right. So, and there's some other changes as well that are thought to be in the same area in the 3-dimensional structure of the polymerase that achieve the same thing. So why is this? Why is it that you need to have, in order to replicate well in human cells, why do you have to have this single amino acid change?
Right. The burning question is there some cellular factor that is inhibiting the polymerase that you have to change to avoid, or is there some cellular factor that helps the polymerase that you have to change to compensate for?
Right. That's a classic question in host range, and it's the classic approach is to take the 2 cells and fuse them and see if there's a inhibitor or something missing. So, Dr. Despommier,
Yes.
if there were an inhibitor in human cells and you fused human and chicken cells, what would would an avian virus be able to replicate in those fused cells? I'm trying to engage you.
It would not. In fact, why don't you ask me what the title of the article represents, the ANP?
No,
what
not
is that?
yet.
I'm just going to tell you what the ANP stands for.
Later.
Okay, fine.
I was going to say I would never put that in the title. I'm just kidding. I would never criticize you, Dickson.
What?
But this is a science paper. We'll get to it, and I'll let you explain it. So you're right. If you fused the cells and there were an inhibitor in human cells, but in fact, this experiment was done previously, and the fused human and chicken cells support RNA polymerase of an avian influenza virus. So
suggests there's something in the chicken cells that helps the polymerase that's missing in the human cells.
Thank you, Alan. I was about to say that
now,
exact
listen,
words.
Dickson.
Yes?
The next thing, which is the basis for this paper, they took advantage of a chicken genome radiation hybrid panel with hamster cells. Now, what the hell is that, Vincent?
Well, I was out here talking with Steve Goff about that, and it turns out that if you blast chicken cells with enough radiation, you can fragment the chromosomes. And if you do that and then you sort of fuse them with normal cells, then you've got a test to see which part of the chicken genome might be operative for particular functions.
So this is a panel of cells.
It's a panel of cells.
They're basically hamsters with bits of chicken DNA.
Correct, and they're all different.
And the panel, I don't know how many panels, 53 different.
It's a clever approach.
Let me just say, in terms of history, because, as Santayana said, those of us who forget history are doomed to repeat it. Okay?
I like that.
I love that saying so much. Did I get it right, Kathy?
Pretty much.
Close enough.
And when I was a graduate student, somebody who I respected greatly in graduate school said, Vincent, it doesn't matter what you've done. It only matters what you're going to do. And I said, well, wait a minute. At some point, you have to remember history. All right. So when many years ago, someone did the same experiment fusing mouse and human cells to find out where the receptor for polio was. And all they could do is localize it to human chromosome 18.
Yeah.
All right.
Huh.
And then many years later, we cloned the gene and it was on chromosome 18, which was cool, but we didn't use those cell lines. All right. So here they take this panel and they simply say, can the avian polymerase work in them? And the assay is very cool because they have plasmids encoding each of the three subunits of the polymerase. They put those into cells and they make the enzyme. And then separately, they have another plasmid that encodes a RNA with a reporter. Okay,
right.
Which I think is luciferase. Yes,
it is
luciferase.
Yes. And for the Calebs out there, by reporter, what we mean is some sort of enzyme that has a very easy assay and no background in the cells that you're talking about. In this case, it's the firefly protein that fireflies use to burn ATP and make light, which doesn't exist in places other than fireflies, and that assay is extremely sensitive.
So a reporter is a gene whose activity we can easily detect.
Yes.
I think a better definition is a reporter is Clark Kent.
Well,
yes.
Super idea.
I'm just trying to emulate Alan. So they do this lovely assay. And again, the luciferase RNA will be replicated by the flu polymerase if the cells are permissive. And they find that 4 out of these 53 hybrid cells support activity.
So I want to make sure I know what's happening
yes,
here.
Sir.
The reason you get a signal is because you're introducing this plasmid. What's going to happen is the plasmid has a Pol-1 promoter on it.
Right.
So that's going to be transcribed into a little bit of luciferase RNA. But then what happens is that if it actually gets amplified by the polymerase, you wind up with a lot of luciferase, actually messenger RNA, because the polymerase makes it. And that's what's happening, right?
Right.
Why don't they make the negative strand?
They must. They do,
so you make,
matter of fact.
Yeah, and then when you make the plus, the message, then you see luciferase.
Yeah, because later on, and I had a question about this too, we can get to it, they assay both positive, negative, and messenger RNA. I want to talk about that when we get there.
Okay. So four of the clones have clearly gotten some bits of chicken DNA that allow the avian polymerase without the adaptive mutation to work in these cells. And they do some gene analysis. They take a DNA array of chicken genes and they hybridize RNA from these cells to it to say what genes are in common. And they can narrow it down to basically 12 genes among these positive cells. 12, that is really good.
Not bad.
That's great.
Not bad.
Yeah, and not only that, but you were talking about history, Vincent.
Yeah, yep, yes.
A long time ago, you would have gotten to this point and said, okay, well, we're done. It's on chromosome whatever.
Yes,
and you can't find anything. Now what do? You sequence all of the transcripts from all of these things, from all of these cells, and narrow it down to 13. That's incredible.
How about that?
No, it's beautiful. Technology makes things go. You're right. It's the future.
And I was wondering if Kathy would have called Jason Long, even though he's in the UK, and asked him the question, what was your aha moment, the big exciting thing? Because I was thinking when he got this result, to such a clean result showing the signal in these few lines above the background, that's a big whoopee, that one.
Yeah.
Mm-hmm.
Yeah.
You know,
And,
amazing result.
yeah, and especially when he does it with, then that takes the 12 individual genes
Yes,
and
right.
finds it.
Yes.
Yeah.
Right.
I was just going to say that using these hybrid cells has been used for many different kinds of experiments over the years. So the human hamster hybrid cells are the ones that came to mind for me, but it's a really nice approach.
Yeah, the human mouse were the ones used for polio receptor. Mm-hmm. So they took each of these genes one at a time and they put them in cells and they ask, is the protein that's made able to allow avian polymerase to work in a human cell?
Yep.
And one of them does, just one.
Exact.
And it could have gone in either way, you know. Maybe none of them, maybe you needed all of them, maybe you needed two of them or three, but one. And now, Dickson, this gene is called CH for chicken, ANP32A.
And the ANP stands for acidic nuclear protein. Number 32A. So that still doesn't tell you what it is, but it's leucine rich and it's got an acidic region and it's got a non-definable non-acidic region as well. Because it's kind of hard to describe it over the
Dickson, does Trichinella
airwaves.
have this gene?
No.
Are you sure?
Well, no, I'm not. Of course, I'm not sure.
Why would you say no? You probably don't know.
I said I'm not sure.
After you said no.
Well, yeah, I corrected myself because you it was a trick question.
Not a trick question.
It's a Trichinella.
Well, actually, it was.
It's a Trichinella question.
Anyway, so this single gene.
So this might be an important protein for other things too. Yes.
Yeah, of course, it's important for the cell, Dickson.
So I think maybe Trichinella might have one.
It might. Why don't you go look now and tell me?
I'll be right back.
Mice have it.
Mice.
Yeah, chromosome 9.
Are you looking up gene cards or something?
MGI.
This has something to do with exporting messages from the nucleus to the cytoplasm.
It doesn't say anything like
I
that.
don't think so.
Here it says transcriptional regulation by chromatin remodeling, mRNA export, and cell death.
mRNA export.
Export. Okay.
See that?
So flu mRNAs have to be exported.
I just said that to prove to you that I've read this paper.
And you do need to prove it.
Well, yes.
Or else you're not getting
Sometimes
your
I don't read the paper.
You're not getting your paycheck if you don't read the paper.
Oh, no, not that.
You know, what if I paid you $500 per episode if you read the papers?
Vincent, now you're talking.
I would pay everyone.
Is everyone listening to this?
If I could, I would. I'd pay everyone.
No, I wouldn't take the money. Trust me, I wouldn't take it. Nah, you can use it for something better, like new equipment.
I'll take his share.
Anyway, this single gene, you put it in a human cell, it allows avian flu polymerase without the adaptive mutation to replicate.
Cool.
And in this clone, this one of the hybrid clones in which this gene is present and it works in their assay, if they knock it down, with siRNA, that abrogates the ability of the polymerase to work. So it's pretty cool.
Yeah.
It's very nice.
Yeah.
Very
All right,
nice.
so
A tight story.
more cool experiments, though. Right?
Yes.
They put this CHANP32A gene into human cells, and they put a couple of different avian influenza virus polymerases in, a variety of strains, and they work. But if you put human ANP32A in those same cells, it doesn't work.
Nope.
So it's just the chicken one, which is perfectly congruent with what we have learned so far.
Yeah.
Now, there is an A version of this thing, right?
Right.
But the chicken ANP32B doesn't work. Doesn't work. It doesn't stimulate the avian polymerase in human cells.
Right.
So the two are different, and we'll find out why that is in a moment. Now, they ruled out effects on nuclear trafficking. So that's not why this polymerase is able to work having anything to do with nuclear trafficking. It's something else. All right, so then they say what happens in human cells if they put this gene, ANP32A, in human cells. It enhances replication. And what else? They go into some RNAs. All three influenza RNA species are increased. So in other words, when you put the polymerase into these cells, you're getting 3 mRNAs encoding the 3 polymerase subunits. Those go up as well as the reporter RNA. And you also get more virus when you put this CHANP32A in human cells. It's got a long name, and it's unfortunate that it's in the title. Now, how about in bird cells? We've looked so far in human cells. Okay, if you knock down CHANP32A with siRNA, you decrease the polymerase activity, and you decrease the yield of infectious virus, which is nice that they did that. Right. What about the human homologs? What are they doing? So they deplete them singly, and either one, if you deplete, it reduces polymerase activity in human cells. So apparently those are needed in human cells as well for replication of polymerase, for activity of polymerases with the adaptive mutation, the 627 lysine change.
So the virus is adapting not to the absence of this protein, but to a different version of it.
That's right.
When it crosses the species barrier.
So the lysine allows it to utilize the human protein, right? And so, and then if you take that away, it doesn't replicate. Now, tell me, I don't know if there's an answer to this. Are there any? Did they look at a human strain without? I guess none of this. No virus replicates without 627K. Is that right? Are there any human strains with 627E? That's what I'm asking. And if so, when you knock down this protein, what happens to it? Do you guys understand what I'm saying?
Yes, I understand. I don't know the answer.
Yeah. I think they said that there are some strains that don't have that, but have clusters of other mutations around the same area. Okay, but they didn't test any of them.
So basically, every human virus has come from a bird at some point. So you have to have some kind of compensatory mutation, right? All right. So these, even for growth in human cells, you need these proteins. Yeah. So what's the difference between the human and the bird? So the ANP32A has an extra 33 amino acids in it, and the B does not. This is so clean, this paper.
Yes.
They're so lucky. Sometimes you get lucky. So they have isolated cloned versions of this gene from chickens, turkey, duck, turducken. Yes. Chicken, turkey, duck, and zebra finch. And these all have the extra sequence. And whereas ostrich, are you whispering ostrich?
I am.
Human, mouse, and pig, they don't have the extra sequence. And the longer proteins increase the polymerase activity in their assay, and the ostrich protein in the mammalian homologs do not. So the short protein doesn't work. And in fact, if they take the extra 33 amino acids out of A, it doesn't work. And if they stick that into the human, it works.
Right.
These are every experiment you would want. I wonder if they were all in the original manuscript. Or
right.
So,
but evidently, this story about ostrich influenza virus was already known to be something different from the rest of the birds.
Mm-hmm.
That's really
what was
cool.
the group of flightless birds called?
Ratites.
Ratites.
It includes ostrich, rhea, emu, cassowary.
Yeah.
I can't read my writing, and kiwi.
Exactly.
Moa and kiwi. Yeah.
Moa.
fantastic.
I think it's really cool that those birds specifically are lacking this thing.
Why is that?
You know? I don't know.
Are they-
It's
Because
so
they
weird.
don't fly? They're more like us?
It is a weird coincidence. Well, they may be. Their metabolism might have adapted to that.
Really?
So do you suppose if we could make a transgenic person with this, we'd be able to fly? That might be good.
No, but we'd probably be tempted to stick our heads in the sand.
But the real
This is a very weird protein, by the way. Like 60 to 70% acidic residues in the C terminus. I mean, it's wild.
But it does lots of different things too, right? So it's
Well, my
a hub
experience,
protein.
whenever I see one of these things that says, we have this protein and we really are not quite sure what it does, and then lists about a dozen different activities, that to me means that a dozen different laboratories have worked on it and discovered, in quotes, that it worked on their thing.
got it. But not necessarily.
And someday somebody will find out what it really does. I probably just insulted about 40 labs.
they're not listening. It's okay.
Okay.
So the real question, of course, is what this protein is doing, and we don't know. But they will now that they have it, they can figure it out. And the other is why does this one gene in chickens have an extra 33 amino acids? What was the selection for that many, many years ago? And I wonder if you could now look at the phylogeny of this gene
Bet you could.
and go back and find a time when it acquired these 33 amino acids.
Yeah.
Right.
And what does that coincide with? Yeah. So that's a study for the paleovirologists, right? Like Harmeet Malik and Sarah Sawyer, et cetera, right?
Yep.
What was it a viral insult or something else, right?
That'll be a paper down the road that you can do on, what is it? What's your TWiVico?
TWiVico, that's a good one. I like
TWiVo.
And Nels came up with, we're going to do a live one in Utah later this year. He said, we're going to do TWiVo in vivo.
That's good.
How cool is that? That guy is very clever.
Right.
Yeah.
I knew this. Anyway, by the way, everyone, tomorrow we're releasing TWiVo number 5 with Sarah Sawyer and Kartik Chandran.
Nice.
Excellent.
Where we're going to talk about this sort of thing we're discussing now, but with Ebola virus.
So if all birds are derived from dinosaurs, as we were led to believe by the paleontologists, when did flightless birds split off from flighted birds?
It must be known.
Archaeopteryx
That must be known,
was
right?
a flight. Yeah, I'm sure it is actually.
I don't know. I thought you would know that, Dickson.
I don't know. I just seen all 3 Jurassic Parks.
So essentially, chickens are a gain of function,
Right.
Yes.
which goes to show that nature is doing these experiments all the time.
Why?
The answer to your question, Dickson,
Yes.
at least from a quick Google search for bird phylogeny, is very, very early.
Oh, that far back.
The kiwis, cassowaries, emus, et al., and ostriches appear to split off pretty much before everything else.
Oh.
So the last common ancestor they had was their common ancestor with all the other birds.
Right.
Penguins, however, are separate.
Yeah, why didn't they throw them in there too? Because they're
Well, they're not just flightless birds. They arose very differently. So they're most closely related to albatrosses, which are sort of the opposite of a flightless bird. They're almost like
they never stop
non-functional on the
flying.
ground.
That's right.
Yeah.
So do penguins have the chicken version or the flightless bird version?
Question.
Don't know. My money's on the chicken version.
Yes.
So, Dickson, you think this is a good antiviral target?
Yeah, I do actually. As long as you don't want to knock it out of the human.
Well, I don't know if that would be bad because you got AMP32B, right?
Yeah, but
And flu likes to have both, but maybe people don't mind. I say we edit everyone's genome to remove this, and now we won't get flu.
And then see what happens.
Well, flu will probably evolve to get around it.
CRISPR will do that.
Well,
Actually, Kathy, wouldn't that be an interesting experiment to make cells knocked out for this and pass flu and see if you can adapt? Would you think that would be prohibited, that experiment?
don't know.
All right, what were you going to say?
I was going to say that I was trying to, when you go into MGI, they talk about they give you references to these genes. And so I think this gene is also called LANP, and it's a member of the inhibitor of histone acetyltransferase complex, which seems to be involved in regulating neuronal differentiation. So I don't think we should delete it from us just yet.
No.
Well, we could delete it from mice. Do they tell you if there's a knockout there in the MGI?
I have to go back to that page. Go back one more click. There are a bunch of mice, 6 phenotypes from 2 alleles and 2 genetic backgrounds. I have to click on this to find out if it's a knockout, but
Like a 3D.
Let's see. Targeted null knockout. Yeah. And I think based on what I saw at the upper level, the homozygous mutants are viable, fertile, behaviorally normal, and show no defects of the CNS.
Bizarre.
Yeah, so the double knockout, the double knockdown here, the polymerase is still 25% active. So I don't know if with virus, if it would be strong enough to do any selection. But
right, and if this is only knocking out ANP32A, maybe you need to knock out ANP32B.
Yeah, right. I think it would be interesting.
It sounds like a grocery store, though, doesn't it? ANP.
Yes,
Yep,
that's
ANP. ANP. Can we put that in the title? So many possibilities here. Anyway, that's a cool paper. All right. Anything else before we move on?
Nope.
All right. ASM is also a second sponsor of this episode, and they would like you to know about a new podcast that they have. It's called Microbe Magazine Podcast. It's a monthly show hosted by Jeff Fox, who is the current topics and features editor for Microbe Magazine. If you are an ASM member, every month you get a magazine in the mail, and it's called Microbe. And he is an editor for that. And each podcast corresponds with an article in the issue, whatever the current issue is. For example, episode number 8 focuses on making yeast strains to produce lager beers with new flavor notes.
Cool.
As if we don't have enough flavor.
Flavor notes.
You can find Microbe Magazine Podcast on iTunes. Just search for it or go to microbeworld.org/MMP. I left the P off on the show notes. MMP, Microbe Magazine Podcast. We thank ASM for their support of TWiV. We have only 2 letters here.
Wow.
The others, we do have others, but they're all requests for proposals. No, requests for
do
this paper or that. So, Kathy, you want to take that first one?
Sure. Dear TWiVniks. Oh, this is from David. What if you thought you sent your child off to college and they ended up on a cruise ship instead? A physician friend of mine has a daughter at Ursinus College in southeastern Pennsylvania. She was one of the early victims of a recent norovirus outbreak. He gives the link to that. Apparently, this kind of thing has happened in the not-so-distant past. For example, 3 different campuses in 2008. And I will insert here, currently, University of Michigan.
Yeah.
Oh, is that right?
Yeah, we had 2 students tell us on Monday they couldn't come to the exam because they were up all night throwing up. And then 2 kids, one kid came late to the class. He said, my stomach didn't feel good. And then one left during the exam for a long time and came back and said, sorry, I was gone so long. I'm not feeling well. And so finally, yesterday, the university has figured it out. But yeah. Okay, so
is this centered in a dormitory or anything like that?
Yeah, a couple of dormitories, but people who don't live in the dormitories, like one of Christiana's students, for example. And so she's at home collecting samples, and Christiana is frantically putting in an emergency IRB to collect samples. I promised her I would announce it to my class as soon as by email as soon as.
Any volunteers? Right. That's great.
So, yes, David, this is even happening as we speak. Okay. It makes one wonder whether college dining facilities like cruise ships need to be much more fanatical about sanitation. Even if they were, the total environment, campus versus ship, is much less controllable, as is the target population. Nevertheless, another opportunity for the TWiV bump. If it takes more than 10 episodes for a podcaster to have a shot at sustainability, how many does it take for a listener? I'm a relative newcomer currently working on 100 episodes or so. I probably won't ever get the chance to make my way through the archive. But for me, TWiV has now achieved the status of ritual, and a very enjoyable and informative one at that. Remember that for humans, the choice is never ritual or no ritual, but rather which ones.
Very good.
That's good.
I like that.
Keep on TWiVing and twimming and twipping and... How many of you remember Robert Crumb?
Oh.
Keep on trucking.
Keep on trucking.
That's right. Zippy, right?
Yep. So this is from
Did he do
Dave.
Zippy?
Oh, sorry.
What?
Did he do Zippy?
I thought so. I may be wrong.
Zap.
What comes to mind?
Zap Comics. Right?
Bill Griffith did Zippy.
Okay.
Anyway, this is from our friend Dave Spector at Professor Emeritus at Penn State.
So Zap Comics were really cool. I don't know if you guys remember. They were just awesome. You remember, Dickson? You have a youth?
I got a ute. Yeah, I had a ute.
A ute?
Are you a ute?
I'm still living my ute.
I mean, My Cousin Vinny is just one of the best movies
I
ever.
love that. I watch it every time.
So David's at Penn State. Now, I'm sad that he thinks he can never listen to all the episodes. I mean, you just could listen to a couple a week.
Right.
Yeah.
People
and
have.
If you listen at 1.5x speed, at least through the weather and stuff, you could do it. Maybe. I don't know. We'll see when we're Professor Emeriti if we want to spend our time listening to the TWiV archive.
Well, Dickson isn't an emeritus, and he helps us do them, right?
So is Rich.
I'm never going to stop until I'm in the ground.
And you won't stop.
And then maybe you'll find a way to do it even afterwards.
Maybe.
Your microbes won't let you stop.
I need to figure out how to keep it going after I'm gone. We have to engage some young people and
Some mute.
build a structure that will keep on trucking.
Yeah.
Yeah, so, Robert Crumb is responsible for Zap Comics, Keep on Trucking, Fritz the Cat.
Fritz the Cat.
Mr. Natural and Weirdo. I think I associate him with Zippy because the drawings are, in my
Similar
mind, at least
style.
kind of similar. Yeah.
Yes, there's a good bio documentary of him. Came out several years ago. It's just called Crumb.
Yeah.
Alan, can you take our other email, please?
Sure. Kevin writes, Greetings, TWiVniks. I'm writing to clarify a couple of points about germinal center B cells and to share a hypothesis about hemozoan and B cell activation. No one said anything incorrect about AID and AID. What does this stand for?
Activation-induced cytokine
activation.
deaminase.
Okay.
Ooh, good.
In germinal centers. But a couple of things might have been misleading. Alan said that during affinity maturation, B cells that don't get higher affinity get deleted. Though this is often the end result, it implies that there's a way to sense an increase in affinity. In fact, germinal center B cells are competing for activation signals, and there's only so much to go around. The highest affinity B cells get the most signal and continue to proliferate. It's like natural selection in miniature, or grant applications. If the initial B cell receptor happens to have the highest affinity even after hypermutation, B cells that retain it would get all the activation signals and survive. The second thing is that you talked about AID in affinity maturation and seemed to imply that sometimes this accidentally leads to breaks in the DNA that lead to translocations. Actually, there are rarely double-stranded breaks in the antigen binding region of the B cell receptor gene where hypermutation occurs. But AID is also responsible for the double-stranded breaks that are intentionally caused during class switch recombination. I haven't kept up with literature since my first year of grad school, but my recollection is that there are large GC-rich switch regions that get excessive C to U mutations that destabilize the chromosome. Finally, Vincent said we don't know why hemozoan would increase AID expression in B cells. I have a hypothesis. Many years ago, Shizuoka Akira's lab showed that hemozoan is a fairly potent activator of TLR9, which is normally thought of as a receptor for DNA. We also know, and he gives a link, that strong TLR9 signaling is able to activate B cells even in the absence of T cell help. It's thought that SLE, lupus, an autoimmune disorder in which anti-DNA and anti-histone antibodies are often detected, may occur due to large quantities of self-DNA getting endocytosed by B cells and aberrantly activating TLR9. Everybody still with us?
Oh, yeah.
Perhaps hemazoin is triggering B cell proliferation non-specifically and or lowering the threshold for activation. Since activation triggers AID transcription or for somatic hypermutation and affinity maturation, maybe they're not measuring increased AID expression by activated B cells, but rather increased numbers of B cells being activated.
Dickson, you know what hemazoin is?
Yes.
Okay. This was about the malaria EBV
Understood.
connection. But I don't remember if you were here for that.
Oh, he was
What?
at the end of
He
that.
came in.
He told us about the stacking enzyme and everything.
Exactly.
And Alan, the reason you tripped over Shizuo Akira's name is because there had been one too many vowels in it.
Yes. Yes.
Okay, so it looks like, are we going to do this next one, Vincent? You said we only
I
have two
hope
more.
so, yeah.
It just came in.
Oh, okay.
Yeah.
But he doesn't tell us how to pronounce it, does he?
No, I didn't. I'm sorry. I was reading my email while you were talking, Alan. Just to see if there was anything that we could add here. An Archan or Arcan. He didn't say
That's okay. I have Twitter open at the same time as this, too. So.
Hi, Vincent. On microcephaly, it's a structural diagnosis, as you know, with presumably many causes. And he gives links to papers about related eye manifestations.
We had thought we didn't know whether eye problems were a common occurrence in microcephaly, and we kind of on the show threw it out to Archan.
And boy, this first sentence of the abstract. True microcephaly is associated with extremely varied ocular abnormalities, the most frequent being squint and optic atrophy.
Yep. Very good. And then on recent genetic mutations and eye involvement, he sends another link to a paper, mutations in PLK4, encoding a master regulator of centriole biosynthesis, cause microcephaly growth failure and retinopathy.
Huh.
Wow. This is why...
Senior author is Andrew Jackson.
Good old Stonewall.
Yes.
Very, very senior.
That's right.
Microcephaly and encephaly involve global growth dysregulation of the brain skull. From an ophthalmic perspective, the brain is an organ involved in processing vision, so it's reasonable to assume that in cases of widespread brain developmental abnormalities, there would be retinal and optic nerve dysfunction as well. I communicate with Tatiana Avsik, the corresponding author on the New England Journal article from last week. They looked at the brain, but not the eyes, for Zika virions genome. Given that the virus is in the brain, it's not a stretch to look for and likely find it in the eyes. Alas, not so easy this time. Thank you. So this is another reason why we should be broadcasting live so people can interact with us. Dickson, we're going to be live in the future.
Excellent.
Are you okay with that?
Sure.
I'll
try to be more lively.
Let's do some pics. Dickson, did you send me a pic?
I did, actually. I sent you two of them, but so I have a choice. This is the 25th year anniversary for Hubble Space Telescope, so I sent some of their best images to this as a pick of the week. And then I ran across a wonderful time-lapse movie made by the Solar Observing satellite, which is about 1 million miles off of Earth and in a geosynchronous orbit with the sun. And it's a movie. I think it's about 6 months of sun activity taken in the ultraviolet spectrum. It's fabulous just to sit there and watch this thing behave, because it's obviously where all the energy from Earth comes from, except from the center of the Earth for those life forms that use that. So I was fascinated by this movie, and I watched a lot of it. I didn't watch the whole thing, but it looks fairly similar, but it's not. It's actually quite wonderful to do.
I feel like this was a pick before.
Yeah,
I
picked a short clip
okay.
of this thing.
I mean, that's fine.
Dixon, you didn't send it to me, though, right, Dixon?
I did. I sent you both the 25-year celebration for Hubble, and I sent you this one. I'm pretty sure I did, at least. If I didn't, I'll apologize over the air. But so I'll pick the 25-year celebration for Hubble. How's that?
Ah, okay.
Oh, here we go. My pick for 377.
Yeah.
You know, I just don't pay attention to your email.
Sometimes you don't.
And so
Be nice to Dickson's email.
That's right.
here
At
we
least
go.
you can be nice to his email, for God's
I
sake.
thought you were going to pick this hydrogen car that goes 300 miles.
No, I sent that just to you because you thought it was fraudulent. I'm sure you do.
No,
but,
if the battery is fraudulent. That seems okay. It's a hydrogen fuel cell. I'm amazed it works.
it's fantastic. 1.5 liters of hydrogen gets you 295 miles per gallon.
But making that hydrogen, Dickson, how much energy does that?
Solar panels. You can split water using solar panels, so I don't think it's going to cost us
And
much.
fortunately, nothing bad has ever gone wrong with a hydrogen-related vehicle.
Exactly. You mean like the Hindenburg?
Yes.
That was not a hydrogen-fueled...
No, that was not hydrogen fueled, and it was also made out of nitrocellulose, which is probably not helpful.
Not helpful as
It
well.
was, really?
Yeah,
And of course, we've never had an accident with gasoline.
right.
Oh my gosh. Kathy, what do you have?
I have something that I came across because of Zika week before last, I think. And it's just, sorry, I was zoomed all the way to the top of the document. Now I have to zoom all the way to the bottom. And it's called...
This is great. This is great.
Yeah,
Yes.
spurious correlations. And so we've picked these before. A listener picked something on 172, and I picked 3 different picks on TWiV 212. And this is just another one from TWiVniks.com. And and it's now a book too.
Yes.
And they have a correlation between United States spending on science, space, and technology and suicides by hanging, strangulation, and suffocation. Or the divorce rate in Maine with per capita consumption of margarine.
Nice.
And they just
I like the number of people who drowned by falling into a pool correlated with films Nicolas Cage appeared in.
Yes.
Yes. and it's not just a single, there's some up and down to it too. Yeah. So whenever people are trying to make spurious correlations, you could just whip out some of these on your phone and show them to them and say,
I think there might be some validity to this. People who drowned after falling out of a fishing boat correlating with marriage rate in Kentucky, but that's
I was thinking that there might be something real too in the total revenue generated by arcades and computer science
Yes.
doctorates awarded in the U.S.
yeah.
That's pretty cool. Yeah, that's very good.
But per capita consumption of mozzarella cheese correlated with civil engineering doctorates awarded.
yes.
It's a lot of late-night pizza. That's all that means.
Rich, what do you have?
What do I have? Nerd alert! Nerd alert! I just cruising around looking up some rocket of some sort, one of these satellites in NASA. I got distracted by the topic of ion propulsion.
These are so cool.
Now,
Good stuff.
apparently, ion propulsion was first proposed in the 100 years ago.
Yeah.
And it's been around for quite a while, but I haven't been aware of it. And so I got a couple of links here, one to a NASA site and another to a wiki site on how ion propulsion works. And basically, this is the propulsion system that's used now on most, if not all, of our deep space probes.
Yep.
And the way it works is you have the fuel is xenon, an inert gas. And it is ionized by bombarding it with electrons that can be easily generated from an electrical source and an anode. And bombarding the xenon in a chamber with electrons will ionize the xenon. So now you have a positively charged particle. It splits off an electron. And then this is passed through a porous grid that has a charge, a potential difference that has so it passes past a positive sheet in the grid and then a negative sheet in the grid and has the effect of propelling the ions at a velocity of something like 90,000 miles an hour out of the engine, okay, and generates a force that way that moves the craft forward.
It's elementary Newtonian physics.
Yep.
Absolutely. I had a physics teacher when I was in college who demonstrated how a rocket worked by, at the end of his lecture, sitting on what was effectively a giant homemade skateboard with a pile of sandbags. And he had the side door of the lecture hall open. And he sat on this thing and chucked the sandbags off the stern of the skateboard, and it propelled him out the door. I'll never forget it.
That's great.
So that's how a rocket works. If you throw mass out the back of this thing, every action has an equal and opposite reaction.
Here.
You propel this thing forward. Now, the reason they do this is that it's so economical. You can only do it on something like a satellite because the force involved is measured in like milli or micronewtons or something like that. It's equated to the force that it is required to hold up a single sheet of notebook paper.
But if you keep applying that force 24 hours a day, day after day, you keep accelerating.
Yeah, in space, right? Right. How fast can you go with these things?
That's right. 200,000 miles an hour. You know, when you apply this thing.
And how long does it take to accelerate to that speed? Do you know?
One place they said 0 to 60 in 4 days.
How about the next 4 days, though? The next 4 days is like what, quadruple?
Oh yeah, I mean it keeps on going, right?
Sure.
The deep space probes carry about one of the ones I was looking at carries 1,000 pounds of xenon, and that will at full power it for years.
Yeah.
Okay. So it's just wild. I mean, to me, it's like science fiction, but it's when you get down inside it, it's fascinating.
So you launch the rocket out of the atmosphere, then you can turn this thing on, right?
Right. Exactly. You can't. This thing will not get you off the planet.
Yeah.
Okay? It's only once you're in space.
So it will never be used for human transport, you think?
Well, no. I mean, it depends. If you want to go to Mars, all you got to do is get into Earth orbit, and then the machine that you're going to use to go to Mars, I don't know if it would be practical for that, but maybe beyond.
Because we're talking about a bigger payload there than these little probes.
That's not necessarily a problem. You could accelerate something, with a slingshot maneuver around Earth repeatedly with nobody on board. And then when it's up to the speed you want, you could launch your astronauts and have them go out and meet it.
Got it.
I'm just throwing that out there. I'm certainly not a space scientist, but
okay.
Every time I cruise around through the NASA site and see all of these things that they're throwing out into space and how cool they are, I just thank my lucky stars that the country has at least enough residual wisdom to keep this kind of research going, because it's just so cool.
And the NASA budget isn't all that much anymore.
No, they do a lot on very little.
I think it's less than the NIH budget, isn't it?
It's true, but they've recently completely assembled the Webb telescope mirrors. So I'm really I shouldn't say anxious, because I'm excited to see the first pictures that come back from that one. That's going to be my pick of the week next time.
So for 2011, the NASA budget was $18.4 billion, and the NIH budget is what, $34 billion or something like. And we complain all the time.
What was the cost of that brand new nuclear aircraft carrier?
Oh, no.
Wasn't that $35 billion
or something like that?
It was crazy. Crazy. I mean, how long do you think it would take it to accelerate that to 200,000 miles an hour?
Alan, what do you have?
I have something that's a much quicker, simpler pick, but a very, very handy item that the CDC just recently released. It's a little quiz. You fill in some information about yourself. Don't worry, it's not too personal. They just ask if you're male or female and about how old are you and some general stuff about your health. And then it gives you recommendations and tells you what vaccines you should have.
I took the test. It says I need seasonal flu, pneumococcal, DTaP, and zoster.
Yep.
And I got them all.
Ooh, look at mine. Hep A, Hep B, flu, meningococcus, Tdap, and zoster.
Lord.
That's because you travel.
Well, don't you travel?
Nah, not anymore. Not outside the United States.
You go to Oregon.
I go to Oregon. I may travel outside the United States sometime, but I don't have, I'm not in the near future.
It's a cool quiz.
I do.
Very
nice.
I
Very
do
nice.
want to travel.
Very handy.
By the way, Alan, I got the kit for the flu swab.
Oh, good.
What is that called?
The going viral or go viral.
Yeah, I got it, but no flu, of course.
Yeah, exactly. My wife got very briefly sick yesterday, and I thought, oh boy, are we going to get the flu? And then no,
maybe you ought to have a flu party, Vincent.
right? Yes.
Yeah, right. Who will bring the flu? One flew over. The bird flu. All right, I'm picking something of my own because I think it's pretty cool. These are my virology lectures for 2016. Cool. Every spring, I teach my course to Columbia undergrads, and I record it. And every year, it's slightly different. And this year, it's in high definition.
Excellent.
Because last year, someone said, I put them on YouTube. Someone said, these are not high enough to zoom in. So now they are high def and full screen and all that. And they change a little every year. This year, I have a lot of new figures from the new textbook. And I don't know, I seem to be more excited this year. One of the students says I'm very animated.
Nice.
So I'm very excited about it. And so
you're alive.
I every year things appear with more clarity in the field. Not everything is clear, but this year certain things have appeared with more clarity, and I hope I transmit that to the students. So I know there are a lot of people out there that know virology. You don't need this, but if you want to learn virology, or if you want to look at a course structure that I think works, all the lectures are there. The slides are there. There are even some study questions. You can find it at virology.ws/course or iTunes University. I'll put a link for that in the show notes.
I'll bet you every year it has more clarity for you, too. That's part of the deal.
It does, for sure.
Yeah.
It does. I look at stuff. Yeah.
So that,
Absolutely. There's some years where things are a little fuzzy and it doesn't really matter. But then the next year I go, oh my gosh, now I get this. And
Right.
that's when you become a better teacher, And it's not just by teaching it, but we work on viruses, and that really helps an awful lot. It translates.
Teaching is always the best way to learn stuff.
Yeah.
Here.
Mm-hmm.
We have a bunch of listener picks from our friend Johnny in Cambridge. For those who would like to step back and take a look before listening to what has happened, the Giant Magellan Telescope on the kids should see this. All right, this is a video. Big telescope with lots of mirrors. It's very cool. And then an understandable introduction to what's going on with sound and gravitational waves at Mashable. Interesting mind-expanding news. Wonder how this might be mirrored at the molecular level of life. And then have these been picks? Award-winning photomicrography of William Van Egmond. Wim Van Egmond. Vim. Have you seen this, Dickson?
I have not.
Yep, it's pretty cool. I don't know if it's been picked, but it doesn't matter. And in case no one has sent these links to you from the Kids Should See This website, what a world, from viruses and infinitesimal life forces to infinity and beyond, and everything in between. The president of Caltech must be proud. And then also a New York Times video on the gravitational waves that Einstein predicted. Show us some links to that as well. So thank you very much for all of those.
I wanted to say one thing I forgot to say when we were talking about the influenza paper, and that is that Anise Lowen wrote a nice News and Views article about it. I thought it was particularly well written and really nicely summarized the paper. And she was the other Anne Palmenberg Award winner along with Nels Elde at ASV. And so I wanted to give her the TWiV bump for this nice News and Views.
Where was that published?
In the front of the same Nature issue.
I thought I looked at that. Let me see where that.
You put it in.
I put a link. Oh, okay.
Yeah, we just didn't mention it. That's all.
Yeah.
Coming home to roost, I think, was the last couple of words of that.
A host protein clips birds.
Yeah.
Bird flu's wings in mammals. Yes.
Stole our show titles.
Right.
Yeah, really good. All right, that'll do it for TWiV 377. You can find it at... Where can you find it, Dickson? Do you know?
All over.
Just don't know. To iTunes and microbe.tv/TWiV. Where can we find you, Dickson?
All over.
All over.
Outside of your office, usually sitting at my desk.
Sometimes sleeping.
Sometimes. Yeah,
you don't
sometimes
deny
I
it,
take
do
a nap. No, not at all. I think an afternoon nap is very therapeutic.
You can send your questions and comments to TWiV at microbe.tv. And I would like to thank all of these wonderful participants. Dickson Despommier, you can find him at verticalfarm.com. Thank you, Dickson.
Pleasure.
Father of the Vertical Farm.
Pleasure. Yummy.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is fun, and I learned a lot.
Rich Condit is an emeritus professor at the University of Florida in Gainesville. Thank you, Rich.
Sure enough, always a good time. Great fun.
Alan Dove, you can find him at turbidplaque.com. You can also find him on Twitter. Thank you, Alan.
And thank you. This is always a pleasure.
And I'm Vincent Racaniello. You could find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.